Today, we're diving into something pretty incredible, a groundbreaking study that's completely changing one of the toughest decisions in cancer care. We're talking about a simple blood test, a test that could spare thousands and thousands of patients from the harsh side effects of chemotherapy. So let's get right into how it works.
Okay, so just imagine this for a second. You've just had surgery for colon cancer. The tumor's gone, which is great, but the anxiety, that's definitely still there. The huge question hanging over your head is, are there any cancer cells left behind? And that leads to an even bigger question. Do you have to go through months of grueling chemotherapy, just in case? For years, that decision has been a bit of a guessing game. But what if it didn't have to be?
So here's how we're going to break it down. We'll start with the huge dilemma facing doctors and patients. Then, we'll get into this new liquid biopsy tech that's designed to solve it. We'll unpack the landmark dynamic trial, from its very first results all the way to the final 5-year verdict. And we'll see what this all means for the future of cancer treatment. All right, so let's really dig into the core problem here. For patients with what's called stage 2 colon cancer, surgery to get the tumor out is always the first, most critical step. But it's after that's when things get really complicated. See, the standard approach has always been to look at risk factors under a microscope. How big was the tumor? Did it look aggressive? Based on those clues, doctors would often recommend what's called adjuvant chemotherapy, basically an extra round of treatment to mop up any microscopic cancer cells that might be lurking. The problem? It's not a precise science, and that means a lot of patients have been getting toxic chemotherapy that, it turns out, they never actually needed.
But that guessing game, well, it might finally be over, all thanks to a revolutionary new tool. Instead of guessing based on risk, what if you could just know? What if you could directly detect if any cancer is still there? That's the entire idea behind the liquid biopsy.
Here's the basic concept, and it's brilliant. As a tumor grows, it actually sheds tiny fragments of its own DNA into the bloodstream. This is called circulating tumor DNA, or ctDNA for short. Now, I love this analogy. Think of it as a molecular smoke detector. If you find ctDNA in the blood after surgery, it's a massive red flag that there's still a fire, active cancer smoldering somewhere in the body. If you don't find it, the fire's probably out.
So this leads us to the billion-dollar question that researchers just had to answer. Could using this ctDNA test as our guide allow us to truly personalize treatment, giving chemo only to those who absolutely need it, while letting everyone else safely and confidently avoid it?
And that brings us to the dynamic trial. This was a huge deal, the first major randomized study designed to see if this whole ctDNA-guided strategy could actually work in the real world for stage 2 colon cancer patients.
The design of the study was actually really simple and elegant. They took 455 patients and split them into 2 groups. In the ctDNA-guided group, the decision was black and white. You test positive, you get chemo. You test negative, you don't. Meanwhile, in the standard management group, doctors made the call the old-fashioned way, based on their judgment of those traditional risk factors we talked about earlier.
And the very first result was a really big one. In the standard group, doctors ended up giving chemo to about 28% of patients. But in the ctDNA-guided group, that number just plummeted, all the way down to 15%.
Let me just put that plainly for you. This new approach cut the use of chemotherapy almost in half. This was a massive, massive finding. It showed that a huge number of patients could potentially be spared all the toxicity, the cost, the anxiety of treatment. But there was still one absolutely critical question left. Was it safe? Did giving so much less chemo lead to more cancer coming back? Well, the answer after 2 years was a resounding no. The recurrence-free survival rates were practically identical. So get this, the ctDNA strategy achieved the exact same excellent outcome, but while using far less chemotherapy. It was just a huge win-win.
But in cancer research, 2 years is just the beginning. The real, true test is long-term survival. Critics, researchers, doctors, patients, everyone was waiting. Would this strategy actually hold up after 5 years? Well, that data has just come out, and it's what we're really focusing on today.
And here it is, the long-term verdict. Let's just look at these numbers. At the 5-year mark, recurrence-free survival was 88% in the ctDNA group versus 87% in the standard care group. And overall survival, which is the ultimate measure, 93.8% versus 93.3%. I mean, these outcomes are just astonishingly similar.
In fact, the difference in both recurrence-free and overall survival rates was less than 1%. This is what researchers call non-inferiority. It is definitive proof that the ctDNA-guided approach is every bit as good as the standard of care over the long haul.
So yeah, the 5-year data really sealed the deal. It confirmed that using this molecular smoke detector to guide treatment is a safe and effective strategy. It allows doctors to confidently de-escalate treatment, sparing patients from unnecessary toxicity without compromising their chance of a cure one bit.
Okay, so this is obviously a landmark finding. But where do we go from here? What does this all mean for the broader field of cancer treatment?
Well, first, it's really important to understand why these results are so powerful. The dynamic trial was a randomized study. That's the gold standard of evidence. It now has mature, 5-year data, and it provides definitive proof that a personalized, biomarker-driven approach can really truly work.
Now, of course, no study is perfect. We should point out it had a relatively modest sample size. It didn't formally measure the impact on patient quality of life, although we can pretty safely guess it was better with less chemo, and it was conducted only in Australia. These are all areas for future research to build upon.
So what's the road ahead? Well, the next logical steps are to test this strategy in higher-risk patients, like those with stage 3 colon cancer. For that small group of patients who do test positive for ctDNA, we need new trials to see if more intensive therapies can improve their outcomes. And future studies absolutely have to include patient-reported outcomes to really quantify the benefits of avoiding chemotherapy.
Ultimately, what the dynamic trial represents is a true paradigm shift in oncology. It's a move away from treating a statistical risk based on what a tumor looks like under a microscope, and it's a move toward treating actual detectable molecular disease that we can find with a simple blood test.
And this brings us to our final thought. For decades, just in case has been a guiding principle in cancer treatment, and it's led to so much overtreatment. With technologies like cDNA testing, we are finally getting the tools to be more precise, more personal, and frankly, more humane. The question is no longer if this will change clinical practice, but how far and how fast this new era of precision medicine will go.